All right, so what I wanted to do, and hopefully this won't be super long, but I just wanted to send you guys a video that was essentially what we would have done in class if I'd been there. And, I'm mostly just sort of interested in recapping some things, making sure that some things made sense. So you don't necessarily have to listen to this whole thing if you felt like you did it and it made a lot of sense. But just I'm going to highlight a couple of things if people do want to talk about this. So actually the one thing, let me show you, what I would like to sort of do is put some of the molecules that we've been looking at into, and I think this has been a helpful way for me to think about this, but basically most molecules in your body, so when we're organizing organic molecules, and it's always helpful to notice that organic molecules are molecules with carbon, but specifically molecules with carbon and hydrogen. And yes, they're the molecules that are in living things. You can also generate them in a lab in many cases, but they're these carbon-based molecules that make up living things. So if you went into a cell, and you can even think about like that video that we watched in June about the cell, pretty much everything in there is one of these four kinds of molecules. And so what we'll do is, so you guys actually probably have done most of these. I'm going to give you, sort of what would be the most common types that you might be expected to know. And so actually what I might do first, so if we even go back to, hopefully you can still see this, but so the first part A of the molecular visualization, a lot of what you were looking at there were the smaller subunits. And then we talked a little bit the other day about how do you build a bigger subunit from a smaller one. So in this case, we looked at like sugars like glucose and fructose, and then we'll also see that this condensation reaction applies to making triglycerides, and we're going to look at this other molecule today. So condensation is going to link molecules together by removing a molecule of water, and then the opposite way, if you want to break down cellulose or amylose or amylopectin into glucose, hydrolysis is the reaction that will do that. So what were we doing? Okay, this chart. So in terms of the simplest subunits, and we basically have these four classes of organic molecules. And this honestly would be a really great, take a full page of your notebook and make this in there. And if you want a snapshot of what are most of the kinds of molecules that I need to know about. But you might want to not make the whole chart first, go through and leave enough room for carbohydrates, and then just add on categories as you go down. But really interesting that, carbohydrates are not a large percentage of our body weight. They're this short-term energy source, but they also can be used for structure. And this is talking about us. Like a plant has way more, has a really, really significantly higher amount of carbohydrates. But the three common, so a monosaccharide, mono is this one unit, the simplest unit that you're going to build bigger molecules from. So glucose is the most common, and there's alpha and beta. And then fructose is the other common, and fructose is the one with 5 in that ring, or 5 atoms, but 4 carbon atoms in the ring. And then galactose, which was really similar, just a different thing going on in carbon 4 with one of the hydroxyl groups. So these are probably most of the monosaccharides that you'll have to know about. In this slide, so actually it's a good example of a condensation reaction between two monosaccharides will give you a disaccharide. And there's just 3 disaccharides that you should kind of have a working knowledge of. And so coming back here, right, so you can take monosaccharides, put 2 of them together, and then the common disaccharides are maltose, which is just glucose and glucose. And then the other ones, so you've got sucrose and lactose. And so these common disaccharides that we talk about, you can just remember that they're all glucose and something else. So maltose is glucose and glucose. Sucrose, I always think about this is literally when you eat sugar, like the white stuff that you cook with or whatever. that is glucose plus fructose. So if you combine those two monosaccharides, you get this disaccharide of sucrose. And I sort of think of like, it's like the fruit sugar or, like the stuff that you get in sugarcane. And then lactose is the sugar found in milk. And it's kind of easy to remember. It's glucose plus galactose. Galactose. It's got that in the name. And then, so hopefully, this makes sense. And then these generally, monosaccharides and disaccharides, are considered to be sugars. Polysaccharides, we wouldn't call them sugars. But they're also composed of monosaccharides. And as you've probably seen, the really common polysaccharides are made of glucose. And so just to kind of put these in from your part B of your little assignment, so you've got so starch is made of amylose and amylopectin. And you might get asked about the differences between those structures. We'll look at that in a second. Glycogen is, and we'll look at the characteristics in a second, and then cellulose. There are other polysaccharides. You just don't end up talking about them all that much. So these, and again, might seem like a lot now, but it's not that crazy in terms of remembering these specific examples of monosaccharides, disaccharides, and polysaccharides. So then coming down, because you guys took a look at some lipids as well. The big thing about lipids is that they're hydrophobic, or like mostly hydrophobic. And when we look at phospholipids, we'll see that there is a distinctively hydrophobic part, and then this one little part that is polar or hydrophilic, and that ends up being a big deal when we determine the properties of structures made of phospholipids, mostly the cell membrane. So when you talk about, so you looked at a triglyceride, and then what we looked at the day before was that a triglyceride is made of 3 fatty acids. And then that glycerol is actually a carbohydrate. So it's this carbon, oxygen, hydrogen. A phospholipid is going to be We're going to look at these in a second. 2 fatty acids plus glycerol plus a phosphate. And guys, I know that it seems like you're just memorizing molecules right now, but we use these all the time when we talk about, so just trust me that eventually this will become more second nature. And then a steroid, I just want to remind you guys, a steroid it's not a bigger molecule. It's not made of subunits. It just happens to be this molecule with these 4 hydrocarbon rings. And so cholesterol is like the most common example of that, but there are also some, so I'm just going to write example here, it's cholesterol. And then I might put a little note in here. 4 hydrocarbon rings is the characteristic of a steroid. Okay, so we're going to leave proteins for now. And we're just going to sort of look at the structures, some of the things that you looked at, and just maybe some common questions. So I'm going to just sort of put up here things that I think you should take away from each one of these molecules. So sometimes you'll get asked about, so they're all made of glucose, except these 3 are made of alpha glucose, and cellulose is made of beta. You probably noticed that if they have a branch coming off, and this is sort of, this is basically like a line, even though it has this twisted helical shape, and then there's this branch point where you have another coming off. So amylopectin and glycogen, and honestly, if you showed me either one of these, I wouldn't be able to tell. Glycogen usually is more branched than amylopectin, but you can't really tell just by looking at them. So 1,4 linkage, you can probably see it the best in cellulose. Somebody was asking about this. So in terms of labeling the carbons, so you got, this is the number 1 carbon, number 2 carbon, number 3 carbon, number 4. And so that 1,4 linkage is between the 1 of one molecule and the 4 of another. And so if you can only make 1,4 linkages, you're going to have a straight molecule. If you can make a linkage between, so this is carbon 6 and this is carbon 1, if you can make that linkage, then you can have a branch. So coming back here, I'm going to go forward. But yeah, so looking at, if you can make 1,6, you're branched. If you can make 1,6, you're branched. And then starch is not branched, it's linear, but it's has this kind of helical or spiral structure. So I just want to go really quickly. We're going to look at these molecules, this is cellulose. And then I want us to try to figure out, we're going to try to figure out what carbon is that, like what number, and don't kill yourself if you don't beat yourself up if you don't know this, but so which one is this, and then which one is this? And the way that you can tell that is if you always look for this thing sticking out, that's the 6th carbon. And so actually this one's probably easier for me to do. That's the oxygen, right? So you're going to go carbon 6 on the left, oxygen, and then clockwise. So this is 1, 2, 3, you probably can't see any of this, but 4. So then over here, so if that's the 1, you automatically will have this idea that that's the 4. But you can also think, this seems kind of weird because we're looking at this like backwards. But so here's your 6, there's your oxygen. And so you're going to go around away from the, and it doesn't look clockwise, but if this glucose were in the same orientation, we'd be going around, starting from the oxygen and then going around this way. So that's what that 1,4 linkage is all about. Okay, just really quickly, something to notice about cellulose, because it's so straight, what you get is that cellulose can kind of pack together and hydrogen bonds will form between molecules of cellulose. And I was trying to find a good image of this. But if you look at, well, both of these, you should be able to see where the hydrogen bonds are forming between. And so here you can see like that red is an oxygen, and then you've got this hydrogen, oxygen, hydrogen, hydrogen, oxygen. And so in glucose, which is a polar molecule because you have these atoms with really different electronegativities, you're going to get hydrogen bonds, and you can see them here specifically going between oxygen and hydrogen, and shown as a dashed line. And the place that this comes up is a cell wall of plant cells, is very rigid and tough and durable because of cellulose, and that you have all these hydrogen bonds holding the molecules together. Okay, so last two for now. So you're going to notice that this looks a lot like, well, this is. So yeah, the end of this, so this is the bond between the fatty acid and this little molecule here is glycerol. And it started off, I should show you guys a condensation reaction to form a triglyceride. But you're going to have the end of this fatty acid, so the COOH, and then you're going to have the end of this O with an H on it. And then to make that bond, you will take out a molecule of water. So that actually might be worth looking at. But so you've got your 3 fatty acids, and essentially you have places for 3 things to bind these oxygen atoms. And so triglycerides, right, they're mostly just fat. You know, it's like when we have fat stored, it's in molecules of triglycerides, and really, really hydrophobic because even though there's some oxygen molecules in here, this huge thing is going to be not attracted to water, and which is why, you know, fat is not water soluble. So here, if you look for the glycerol, you're going to see it right here. And then so you just end up having 2 fatty acids. And then that third space is actually, so this is a phosphate, PO4. And then this thing, oftentimes there's a choline on there, like this extra. Honestly, I wouldn't really worry about that. In my mind, whenever you hear about a phospholipid, it's 1 glycerol, 2 fatty acids, and a phosphate. And then when you look at it, what you're going to see is that this section is, we're actually going to say that this is not hydrophobic or hydrophilic, but it's something that we call amphipathic, which means it has a hydrophobic part and a hydrophilic part. But we're going to get to that a little bit later with cell membranes. Okay, so that, yeah, I think I'm going to do this later. Yeah, let's come back to this. But I just wanted to recap that. And if you can start, kind of organizing, and then we'll add proteins and nucleic acids a little bit later.